Good morning, everybody, and thank you very much to the organizing committee for inviting me to talk to you today. And it's a great pleasure to be here. And it's a very exciting time to talk about artificial pancreas research, and hopefully I'll convince you in the next 20 minutes that Europe is in par or if not a little bit advanced in artificial pancreas research, in single hormone research at least. There have been a large number of studies conducted in Europe, so I won't have time to go to details of all the studies, but I'll try to give you a broad overview of the published studies, and I try to be inclusive as possible and include work from all the different groups in Europe to give you the breadth, some idea about the breadth of work in Europe. I have no commercial conflict of interest, but I was fortunate to work in this field for the last 5 years or so with the Cambridge Artificial Pancreas team, and through that also the AP at Home Consortium. I will divide my presentation into several areas depending on the study environment. I will start with true home studies, where the study was done under complete free living conditions without anybody following or telemonitoring, etc. And then going through transitional studies to research facility studies, and a few words about psychosocial assessments, and conclude with a summary. So starting with true home studies, so the Cambridge group has published 3 studies using their model predictive control algorithm under free living conditions. So these studies were done either single center or European multi-center trials. All 3 are published, as you can see. All 3 studies use the Florence prototype, which was sort of developed in Cambridge in conjunction with the device companies. So this system uses the Navigator CGM system, which is attached to a small bedside computer, and then it sends a signal to the Dana pump every 12 minutes or so to control the pump. And this was the first prototype developed for these studies. And all 3 studies adopted a randomized crossover design where each participant underwent both arms of the trial, and there was a variable washout period. We provided training for them at the beginning of the closed-loop arm, but after that the study was conducted in their home setting without any direct supervision. We had a 24/7 telephone line for any problems. This slide summarizes the characteristics of the three studies. The first study, the APCAM 06 trial, was 16 adolescents, and this was a single-center study for overnight closed loop for 21 days, and this was funded by JDRF. And the second study, Angela 03, was a UK multi-center trial in Cambridge, London, and Sheffield. 24 adults, 28 days of overnight home closed loop. And finally, third study, the AP at Home 2, was a hybrid day and night closed loop trial. This was European multi-center in Cambridge, Neuss in Profil Institute, and Medical University of Graz under the AP at Home consortium for 7 days at home. So this is the results, summary results of the first trial from the adolescents. So as you can see on the table, the time in target between 3.9 to 8 was significantly higher with closed loop. The mean glucose was lower, but this improvement predominantly came from reducing hyperglycemia. There was no difference in the hypoglycemia, but the number of nights with at least 20 minutes of hypoglycemia less than 3.5 was less with closed loop. And the algorithm had to give more insulin to achieve these results in this adolescent study. And this is the summary from the adult study. This is for 28 days at home, and as you can again see, very similar results. Time in target much higher with closed loop, lower mean glucose. Again, this is predominantly from reducing hyperglycemia. Hypoglycemia was low in both arms, and there was no difference with closed loop. And once again, algorithm needed more insulin to achieve these results. And finally, the third study, the AP at Home 2 trial, this is day and night closed loop, hybrid closed loop. Once again, the closed loop increased the time in target, reduced the mean glucose, and again, this is from reducing hyperglycemia. There's no difference in hypoglycemia, and the total daily dose there was a tendency towards low in closed loop arm. The next home closed loop study comes from the DREAM consortium, which is a consortium established in Slovenia, Germany, and Israel. So they use the MD logic system. This is an algorithm based on the fuzzy logic theory. It tries to imitate the decision-making process of a diabetes doctor. It utilizes individuals' data to personalize the algorithm. It has the ability to learn, alerts modules, and remote monitoring, etc. So they have published a number of studies as well, looking at the feasibility and safety of their studies as listed here. But this trial I want to show you is the longest home study published to date. So this is for 6 weeks at home in adults and children. This was a single-center trial. Again, randomized crossover design using Medtronic devices. And they looked at, and I show you the results in the next slide. So as you can see, the time spent in hypoglycemia was lower with the closed loop. Time spent in the target range was much higher, less hyperglycemia, lower mean glucose, and this was all achieved with giving slightly lower insulin compared to the control arm. So truly impressive results, improving all parameters at home setting. The final trial under the home category, this is from Dr. Divry's group in Amsterdam. So this was our only sort of dual hormone study. This is a feasibility trial for 48 hours at home. But unfortunately, they had some technical issues, so they were only able to analyze 11 patients. The first 12 hours was at the clinical research facility, and then the next 48 hours at home. They used a PIG algorithm, again Medtronic sensor, again fully closed loop without meal exercise announcement in this study. There was a tendency towards improving time in target, but didn't quite reach statistical significance. The median glucose levels were lower with closed loop, but this unfortunately came at a slightly higher time spent in hypoglycemia. Then I move on to the transitional hotel camp studies. So this study is from our colleagues in Italy, once again conducted under the AP at Home consortium. This was a feasibility trial in a single-center trial in 6 participants. They were admitted to a hotel in the evening, and they conducted first 14 hours in the open loop, followed by next 28 hours using the closed loop system. They were free to move around the clinical facility and its vicinity. They use a modular model predictive control algorithm specifically to assess the postprandial control. They use the meal control, meal bolus, using patients' usual insulin to carb ratio plus the predicted glucose levels. What's shown here is the blue bars are the time spent in the target range in the 4 hours after each meal. And as you can see, the first meal, the dinner meal under the open loop, about 68% in target, followed in the blue lines after dinner was about 94% with the closed loop. And you can see the breakfast is the most difficult meal to control, again, much lower time in target than the dinner meals. In the next study, they went a step further. So this is a multi-center trial in Amsterdam, Montpellier, and Padua. They use the DIAS system, again, Dexcom G4 sensor, and the Roche pump in this study. So similar design, admitted to the hotel in the evening, the first dinner and the overnight and open loop, followed by closed loop control. And when they compared these two periods, they found the closed loop reduced the time spent in hypoglycemia, and at the same time increased the time in target from 62 to 83%. I think many of you are familiar with this study. This was published in New England Journal from DREAM Consortium. This was conducted in a diabetes camp. There were 2 consecutive nights, a randomized crossover design, either closed loop or the open loop arm. And the primary outcome on this study was hypoglycemia, again MD logic system, and as shown here, the number of hypoglycemic episodes was significantly lower with the artificial pancreas system compared to control, and at the same time they increased the time spent in the target range. So it's under challenging camp conditions. Finally, the third category is the clinical research facility studies. Now, there are lots of studies under this category from lots of different groups, but I've selected 3 studies from groups which I haven't mentioned so far. So the first study is from Dr. Hernandez's group from Spain. They used a predictive rule-based algorithm which consisted of a control-to-range module and a safety control module. It was a hybrid system with a partial pre-meal bolus, about 50% bolus, and randomized crossover looking at overnight and post-breakfast control. So the results are shown here. The time spent in the target 3.9 to 8, significantly high with the closed loop, and that's one of the highest I've seen, 95.8%. And as you can see on the profile, the dark gray area, which is the closed loop, very narrow variability and very tight and uniform control achieved in this trial. And this is from our colleagues in Imperial College in London. So they're working on a system, an algorithm based on the mathematical model of beta cell physiology. This is implemented in a miniature silicon microchip. So this first study was a non-randomized feasibility trial. They did a fasting study and then an overnight study. They also used the N-light sensor and the AccuCheck pump. So you can see on the table, under fasting conditions, pretty good time in target in both narrow and broader target, but with overnight and post-breakfast, there's some improvements are required to the algorithm to improve the time in target, which I understand they're working on, and they finished further studies. Finally, this is from Dr. Nogard's group in Denmark. So this is also model predictive control algorithm, 2 feasibility studies conducted in the overnight period. The first one contrasting closed loop and open loop. Second study, closed loop in the starting at normal glucose and high glucose. And once again, they showed less hypoglycemic episodes with closed loop, and the time in target increased. So far you saw all the biomedical outcomes, but what about the psychosocial outcomes? So as you know, the AP systems does have certain demands, and whether the patients with type 1 diabetes can meet these demands is an important question. They will have to carry additional devices. It will increase the visibility of their disease. There is increased alarm burden. Unfortunately, the problems with connectivity, calibration, sense error still happens. So these are important issues, and it's important to have realistic expectations in our patients that it's not going to sort out all the problems, but they may be able to accept a trade-off for the good control. And this study here is from Cambridge Group, where they conducted interviews in people who've done the overnight closed loop for 21 days. They conducted semi-structured interviews with both adolescents and their parents. And what they found was they had a positive impact closed loop on the improved sleep, reduced anxiety, stable glucose levels, and they felt safe. But usual things like alarms, uncomfortable, discomfort, calibration issues were the negative themes. So there is still some way to improve those things, and they are improving all the time, as you know, but there still be some issues to think about. So there are, again, large number of studies currently happening. So there are 2 to 3 months overnight studies happening in adolescents and adults, both in UK and European multi-center trials. There's a study in pregnant women over 3 months in UK. And if you look at both day and night closed loop, we started the 3-month day and loop closed loop study and the AP at Home Consortium, and this is about to finish in May this year. We're looking at that, to look at those results. Very excited. So, and there are plans for UK and US multi-center trials in larger number of patients over the next few months to start. and none of these studies will be feasible without the generosity of funders. So I thought I must mention one slide at least about our funders. So the AP at Home Consortium, a European Commission, has donated large amount of money for this research. JDRF and the various bodies in individual countries, NIH, etc., and also our industry partners. So, to summarize, in Europe we have studied a variety of patient groups under diverse clinical conditions. These include inpatients, hotels, camps, and home studies. We've also done studies in very young people, type 2 diabetes, critical illness, and pregnancy. I haven't shown them today due to time constraints. We have a variety of different algorithms, the model predictive, MD logic, bio-inspired, controlled range, etc. And compared to current best treatment, European closed-loop studies have shown significantly better overnight time in target in both research facility and at home, reduced hypo burden in the research facility and at home, and also better daytime glucose control. The longer studies, which give details about HbA1c results, are due to complete, and they will give us further information. So I like to finish by saying Europe is leading the way in single hormone closed-loop research, and I'd like to thank my Dr. Horka and my colleagues in Cambridge and colleagues in the AP at Home Consortium and several other members, and thank you very much for your attention and staying till the last talk.